So in this lesson, we're going to be focusing at how we get all of the different types of genetic variants that we find as a result of sexual reproduction. So what exactly happens during meiosis to make it so that we end up with different combinations of genes and offspring produced through the process of sexual reproduction? So we know that during meiosis, there's three major ways that it actually creates genetic variation. So the first major way that we get variation is from the process of crossing over. So remember that crossing over occurs during prophase 1 of the first division during meiosis. And this is going to be what happens when we actually get the physical exchange of genes between the maternal homologue and the paternal homologue, so that we end up with unique combinations of genes from mom and dad that end up going into the gametes when we form them. We also look at the process of independent assortment as another way of creating diversity. So independent assortment actually comes into play during metaphase 1, when the homologous pairs of chromosomes actually line up at the equator. Now, what the law of independent assortment states is that the positions or the orientation of the homologous chromosome pairs is completely random. So whichever homologous chromosome you get is going to be a matter of chance just based on how they line up. So essentially, in this case, if we look at it here, one possibility of how they line up is that both of the chromosomes from dad are on one side and both the chromosomes from mom are on one side. So that when they separate, the gametes can either have the majority of their genes coming from dad or the majority of the genes coming from mom. Or they could have lined up so some are from dad, some are from mom on one side, and all vice versa. So when they separate, you get a nice equal mix depending on which gene from which parent you end up with. So again, really which version of the homologous chromosome pair you get is due to matter of chance. And then thirdly, we look at also random fertilization. So just by the process of meiosis, which one of the millions of different types of unique sperm that dad can create just so happens to fertilize which one of the millions of different types of eggs that mom can create is also completely up to chance, right? So which egg decides to start maturing during that ovulation cycle and which sperm just so happens to be the fastest that day is all going to determine which genes or which combinations of alleles that you inherit. So we're going to start focusing a little bit more on crossing over and how that actually happens within the cell. So remember, crossing over is really just when we exchange genetic information between the chromosome from mom and the chromosome from dad when the homologous chromosome pairs are aligned with each other during prophase 1. And so another term we typically use to describe crossing over is as genetic recombination, because we're recombining genes or alleles from both mom and dad. So you end up with a nice mixture between the two. So in this case, whenever crossing over happens, the sister chromatid that ends up with containing genetic information from both mom and dad, we refer to as our recombinant chromatids, versus the chromatids that stay the same, so there's no shuffling of genes on them. We refer to them as our non-recombinant or our parental chromatids here. So let's take a look at how this actually happens. Now, remember, during prophase 1, all the homologous chromosomes are going to start to pair up with one another. So chromosome 3 from mom finds 3 from dad, and 10 from mom finds 10 from dad, etc., etc. And the way that they're actually linked together is through a process called synapsis. Okay, and we'll look at that in just a second. But just know that once they're connected together via synapsis, the pair is referred to either as a bivalent, bi because it means two, so two homologous chromosomes together, or as the tetrad, since the word tetra is 4, and we have 4 chromatids coding for the same genes here linked together. Now, in order to actually create synapsis, we do this through the process of creating what we call a synaptonemal complex. Now, the synaptonemal complex, you can kind of think of like a glue or a paste that kind of forms between the two pairs to keep them together. But really, this is just a complex of protein and RNA that kind of acts as like a glue to physically link together our homologous pairs. Now, while they're kind of linked together during synapsis with the synaptonemal complex, is when we're going to start to see the exchange actually occur between different regions of the chromosome, where part of mom's chromosome can actually break off and recombine with dad's, and part of dad's recombine with mom's. Now, typically the only chromosomes in your cell that will actually undergo synapsis are going to be your autosomes, right? So remember, autosomes are going to be the non-sex determining chromosomes that just contain genes for your traits. Now, since they're going to be the autosome pairs are going to be identical, it's easy for them to link up because they contain info for the same genes. But if you look at the sex chromosomes, it's going to depend on whether or not you got two X chromosomes and are female or one X and one Y. Now remember that the X and the Y differ in size and they don't code for the same genes. So it's going to be really hard for an X and a Y to pair up and shuffle genes because they're not the same chromosome. So usually it's only going to be the autosomes that will come together and actually recombine during the process of synapsis to do this. However, if you are female, sometimes it may happen on the X chromosome, just not as often as with the autosomes. So again, while they're in synapsis, we can see this breaking off of pieces of the chromosome and a physical exchange between mom's chromatid and dad's chromatin to shuffle the genes. So the point of crossing over, or this little X that we see form here between the two chromatids, we call this a chiasma, okay? If there's multiple chiasma occurring on the same chromosome while they're in synapsis, we refer to that as chiasmata. That's just the plural for that. And these chiasma, or the formation of these chiasmata, leads to the recombination of your alleles, okay? Now, another thing just to note is that recombination only happens between non-sister chromatids. So what that means is that it has to take place between a chromatid from mom and a chromatid from dad. If recombination happened between identical chromatids, you'd just be shuffling the exact same identical gene, and that would be useless, right? No shuffling really happening there. So crossing over only happens between non-sister chromatids whenever we form this chiasmata or chiasma to do this. So just take a look at the diagram, see if you can pick out how many chiasma or points of crossing over we actually see here in this chromosome. So pause it for a second, see if you can find it. So here the answer, we see 1 point of crossing over here, so that's 1 chiasma. Another point of crossing over here, that's 2. Another point here, another point here, and another point here. So we've got 5 actual chiasma here forming where we're recombining genes between the maternal and paternal chromatids here while they're joined together. Now typically the average is around 2, so 2 chiasma per bivalent or per tetrad, sometimes a little bit more than that, but here we can see 5 actual physical points of contact and crossing over. So let's try another one. Pause it and see if you can find how many chiasmata here are forming between the two chromatids. So here the answer is 2, right? So 1 point of contact or an X forming here, and then 1 point of contact or an X forming here. So 2 total chiasmata per this chromosome pair while they're in physical synapsis, okay? So when we look at the amount of genetic diversity we can actually get from crossing over, if crossing over were to happen an average of 3 times per meiosis, we would end up with over 67 million different possible genetically different gametes that we can form just by the physical crossing over during meiosis. So this is a huge source of genetic diversity that occurs during prophase 1 when the homologous chromosome pairs line up. Now just know whenever you have to, if drawing a diagram of crossing over, you're going to want to make sure you use 2 different colors, one to denote the paternal chromosome and one to denote the maternal chromosome. And note that crossing over happens between the ones that are actually physically connected on the inside, because crossing over has to happen between one from mom and one from dad. So that at the end, right, the ones at the outside that don't actually undergo or form the chiasma are going to be the same, so no differences in genes. So one from totally mom or totally dad, one containing the mixture, and then one from totally mom here on the outside. So we only get the crossing over where they actually physically overlap. And again, remember the ones containing a mixture of the two we call the recombinants. So recombination has happened to shuffle genes. The ones that don't have anything shuffled are just the normal paternal chromosomes. So just again, another way of showing the recombinants here and how to pick them out, right? So the ones that are normal at the end are going to be the ones that contain just the combinations of genes you found in the parents, right? So here we know the parents, dad's going to give you a big A with a big B. Mom's going to give you a little a with a little b. But during crossing over, some of the sister chromatids on the inside can exchange them, so you end up with a new combination that was not found in the parents. So without recombination, we would have never seen a little a being inherited with a big B, or a big A being inherited with the little b. So our recombinants are going to be ones that have new combinations of alleles that were not found in the actual parents there. So now let's take a look at Mendel's law of independent assortment. Now remember, Mendel came up with all of his laws before we really even knew DNA or genes and how that worked, or even how meiosis occurred. But in his law of segregation, what he essentially stated was that each parent contains two alleles or two versions of their genes, and they're going to separate during meiosis so that each egg or sperm is only going to contain one. So we know that the alleles are going to separate, so each parent actually contributes equally to the formation of the offspring. Now, in his law of independent assortment, what he said by this was that during the segregation of alleles, the alleles are going to separate from one another independently from other genes. So what this means is that the inheritance of one gene or trait is going to be completely independent or separate from your inheritance of another gene or trait. So which allele you end up in your gamete is going to be completely random and independent of other alleles. So for example, let's just say that I have long hair. Okay, so what Mendel would say was that if you get long hair, there's nothing that states that you also have to inherit light skin or blue eyes. If you inherit long hair, you can also inherit dark skin and green eyes, or you can inherit kind of like a medium tone skin and brown eyes. There's all different types of combinations of traits you can get. So there's nothing that states that if you get long hair, you have to also get light skin and blue eyes. You can get combinations of other genes and other traits along with that long hair. Okay? And then so in this case, like we say, big A can be inherited with big B, or big A can be inherited with little b, so you can get a different combinations of traits there. Or little a can be inherited with big B, or little a can be inherited with little b, right? So all different types of combinations. So which allele or which trait you get is going to definitely be separated independently from other genes. Now, and this is going to be due to the random alignment or how the chromosomes actually line up during metaphase 1 of meiosis, right? So if we come back here looking at our possibilities again, we can see all of dad's copies can be on one side and all of mom's copies can be on the other, or we can have a mixed match of each. And so depending on how they line up and segregate during meiosis 1, remember meiosis 1 separates the homologous chromosomes, meiosis 2 separating sister chromatids, is going to determine which combination of gametes you have. Now, this is a really simplistic version because it's really only showing the inheritance of 2 chromosomes. So if you look at us, when we have 23, there's a ton of different types of alignments where we can see, based on just randomly how they happen to line up or orient themselves at the metaphase plate to be separated. So this means that an allele on one chromosome has an equal chance of being paired with or separated from any other allele on another chromosome. So their inheritance is going to be completely independent of each other if they're on different chromosomes. And here's an animation for you if you want to pause and take a look at that. But like I said, this really only holds true if the genes are unlinked, meaning they have to be on different chromosomes in order for independent assortment to work based on how they line up at metaphase, right? So if this is chromosome 1 and chromosome 2, they can line up in different orientations during metaphase 1. So big A can be inherited with big B if they line up on the same side, or big A with little b if they line up on the same side, or little a with big B if they line up on the same side, or little a with little b if they line up on the same time, to get all these different combinations here. However, if the genes are linked, meaning they're on the same chromosome, they're going to always tend to be inherited together. So for example, gene big A is on the same chromosome as big C. So typically these would be inherited together once they line up during metaphase 1 if they weren't separated during genetic recombination. So this only holds true for unlinked genes or genes on separate chromosomes, because if they're on the same one, they're going to be inherited together. So to determine the different types of, or the different amounts of possible genes you can get from independent assortment, we use the formula 2 to the n to describe the possible orientations we can get. So because we have 23 chromosomes, the possible different combinations of gametes we can get just from independent assortment is 2 to the 23rd, meaning we can form over 8 million different gametes just based on how they line up during metaphase. So due to crossing over an independent assortment, there's never going to be another you.